Hi guys, I'm back. So chapter 13 part 3. So now we're talking about multiplication of animal viruses, since we already understood how multiplication of bacteriophages inside bacteria happen. In this case, we're going to look at the phases of the multiplication of these viruses. So usually you have an attachment, and that attachment happens via a receptor. Now try to think. I know you're thinking about the coronavirus, so let's use that just as an example for you to understand how it works. The main contact for attachment for the coronavirus is the ACE receptor, which is the angiotensin converting enzyme. It's an enzyme that is present on the surface of some cells, mostly in the lungs, and that's where the virus connect to be able to enter the lung cells. Okay, so that entrance is always mediated by a receptor, but it could happen through two different mechanisms. So let's just talk about the whole general thing that we're going to talk about here. So after entry, we're going to talk about uncoating, biosynthesis of the nucleic acid and proteins, maturation, which is the assembly, and release is always by budding when you talk about enveloped viruses. Okay, so this is what we have. There are some viruses that are animal viruses and are not enveloped, okay? But we're going to tell you specifically here how enveloped virus exit the cell. So you have the host plasma membrane here, right? And then this is the viral particle, and this is the cell, right? And then through receptor-mediated endocytosis, which is, like I just told you, there are some receptors that the virus has an affinity for, depending on the virus and the cell. And then that is going to create a contact, and then it's going to internalize this virus. Or it can happen by fusion of the viral envelope to the plasma membrane of the host cell. That's another mechanism. For example, the herpes virus uses this fusion mechanism, okay? Once the virus, sorry, this is the release. The release, let's talk about the process and then I'll tell you about the release. I think it's a better order. When you think about a DNA virus, you look into this, right? What happens first? First, you have attachment of the DNA virus to the host cell. Or sorry, to the virus, yeah, that contains DNA, so you call it DNA virus, to the host cell. Entry or an uncoating that can happen by endocytosis or fusion of the viral envelope with the plasma membrane of the animal cell. This is the replication of DNA containing animal viruses. So the virus has a DNA. That DNA, this DNA here inside the animal virus is this one here, right? This DNA goes to the nucleus and makes a molecule of RNA. The mRNA has to go to the cytoplasm, right? It's here now. These molecules are going to be important because they're the template for translating the proteins that are going to make the capsid of this virus. Then capsid proteins go back to the nucleus, you see, to assemble the virus inside the nucleus because that's where the DNA is. Once assemble is ready, maturation is finished, you have the release. During the release, this is what happens. You have the viral capsid here, and this is an enveloped virus. So how is that envelope going to be formed? The virus induces the spikes proteins, which are these little blue points, to be formed and embedded in the plasma membrane. And then you have budding of the plasma membrane of the host cell, and then it just excises. Then you have the enveloped virus. This is an transmission electron microscopy showing the whole process. You see, this orange part here is because of the spike proteins. And then you have initial and end of the budding and excision. Okay? So this is how DNA viruses are replicate inside host animal cell. Now we're going to talk about breathe. It's a similar mechanism when the virus is RNA. Okay, when the genetic material of the virus is single-strand RNA or double-strand RNA. I'm going to focus on one type, which is the positive or sense strand. So every time you see positive RNA, I don't have my pen here today, positive RNA, that's going to mean this is the mRNA. That's not working. I don't know why I'm trying. Okay. See, viral RNA serves as mRNA, messenger RNA, for protein synthesis. What am I trying to tell you here? This is what I'm trying to tell you. I'm trying to tell you that this is a little complicated and I want you to pay attention. Virus is here, right? Virus, let me make this a little thicker. Virus is here with the capsid, with the RNA. What's going to happen to this virus? This virus wants to enter an animal cell. Attachment goes in. Because it's an RNA virus, it does not go to the nucleus. That RNA, so the virus disassemble, everything is disassembled here. You see, the positive mRNA, the viral proteins, which proteins? The RNA-dependent RNA polymerase. It says here, RNA-dependent RNA polymerase. We don't have these enzymes. Can we make RNA from RNA? No, we can't. We can only make RNA from DNA. Viruses have an enzyme that uses RNA as a template to make RNA. That's why they can do it. So they don't need our machinery, except for, they don't need our enzymes, just our machinery to be able to multiply this positive sense RNA. So this sense RNA makes, so this is the positive RNA. It makes the negative RNA. Why do you need to make the negative? Because the negative is the complementary to the positive. And when you make the negative, you can make many positives because it's the template, right? So then this negative one is this one. And this one here is making many positive ones. You see how now you have three positive here, three positive RNAs, because you use the negative, which is the complementary, to make it. Okay. Once you make the RNA, what else do you need to make? From this RNA, the RNA-dependent, the ribosomes can use, the ribosomes in the animal cell can use this mRNA to make a capsid protein. Once everything is made, capsid is assembled, RNA plus RNA-dependent RNA polymerase repacked inside the capsid, and virus exit the cell. Okay, that's it. Hard a little. I think what's hard is here to understand that the positive is the messenger RNA, but you need to make a negative sense, an antisense or a negative RNA to be able to do more positive sense RNAs. Okay, so this is how an RNA virus multiplies. When you think about the negative sense, it's just going to be the opposite of this, and the double strand is going to be very similar to this, okay? So they do not go to the nucleus. Everything happens in the cytoplasm. And then we have the retrovirus. So there is this family of viruses called Retroviridae, and lentivirus is one of the genus of virus, and then you have the HIV, which is the species, right? That belongs to the lentivirus genus. Oncoviruses can also be retroviruses. So how does the retrovirus multiplication happen? So now we have, let me see where is number one here. So we start with virus being incorporated or internalized by the animal cell, right? And then you have the viral RNA here. Remember that this RNA is a different, it's not a different RNA, but it's a different, usually it's in the HIV, it's two identical copies of the same RNA. That's what the genetic material of the virus is. And then that's going to be packed with viral enzymes, right? This virus comes with enzymes. Which enzyme? This enzyme, reverse transcriptase. What does reverse transcriptase do? Transform RNA into DNA. Can we do that? No. The only thing we can do is RNA from DNA. We cannot do DNA from RNA. We cannot do RNA from RNA, right? And then with this, you make DNA from RNA because of this powerful reverse transcriptase enzyme. And then this piece of DNA now can incorporate as a provirus in the genome of this animal cell. And then with this incorporated as a provirus, you can make as many viruses as you can. Okay? So when we talk about latent infection, it could be a DNA virus or it could be an RNA virus that is incorporating into the DNA as the DNA, right? Because it makes the DNA. And then it makes as many copies as it needs, and then new viruses excise and are released from the animal cell, okay? Not necessarily killing it in the process. So viruses and cancer. There are some viruses that can cause cancer. And the way they cause cancer, so we have an oncogene or a proto-oncogene in our genomes. These are genes that could lead to formation of cancerous cells, but they're just there. Why they're just there and dormant? Because there are repressors that don't allow these genes to be expressed. When these genes, when the repressor is missing, these oncogenes get activated and transform that cell that has those genes in cancer cells. How is the repressor would possibly not be there? There are some viruses that inhibit the repressor, and then you can make all these oncogenes products that are going to turn the cell into a cancerous cells. Sarcoma is one type of cancer of connective tissue that can be affected by viruses. Sarcoma di Kaposi is a cancer of connective tissue from vessels that are usually typical in HIV patients or AIDS patients. Those are induced by the HIV virus. Okay. You also have adenocarcinoma that can be induced by viruses. There are one virus that can cause different types of cancer is the Epstein-Barr virus. Epstein-Barr virus can cause different types of tumors, such as Burkitt's lymphoma and some other cancers of throat and nose. The human papilloma virus can cause cervix cancer in the uterus. And there are other viruses that can also cause different types of tumors that we don't need to talk about all of them, okay? Remember what a latent virus is. So the difference between a latent and a persistent virus. A latent virus is a virus sits as a provirus in the genome of the host cell and one day gets reactivated. A very good example is cold sores caused by herpes virus and shingles caused by the chickenpox virus. So a persistent viral infection, the main example here could be measles that could be persistent during a long period of time and cause symptoms during the whole period, or another good example is HIV. So acute infection, for example, flu, right? The flu virus, the influenza virus is going to cause an acute infection like this. Persistent virus, good example, HIV. It persists, it's not very visible for very long, but one day you can see the number of virions increasing and more symptoms appearing. In a latent infection, the virus was there all the time, but one day it gets reactivated, like shingles after somebody had chickenpox, okay, which hopefully doesn't happen anymore with nowadays kids, because hopefully everybody gets vaccinated. But when we were kids, that was a possibility. So when we get older, we could hopefully not get shingles. These are examples of latent infections. It's just a table from the book. And persistent infections. And I just want to quickly tell you about prions. Prions are not viruses. They are proteinaceous infectious particles. So it's a protein that transforms other proteins into sick proteins. So we have a protein in our body that is similar to the prion protein. Once we ingest meat of cow infected with prion, which is not thermolabile, so it doesn't get killed by heat. That's why it's so important that cows get tested for this, because frying the meat, cooking the meat is not going to kill that. So if we ingest that, it can go to our circulation, go to our brain, and transform, our PRPC proteins, which are these ones here, into PRPSC. So the PRPSC is the sick protein. Once it touches our little blue proteins, it transforms it, and then we start to produce these sick proteins because they keep transforming all our PRPC's proteins. With that, this causes this PRPC, which is our functional protein, is involved in a lot of different metabolic functions in the cell that include like when the cell is gonna die and proliferation and other things. So when that protein is missing, you start to have death, current, like super important and numerous death of nervous tissue cells, because that's happening in the brain, and it makes the brain tissue form holes. And that's why one of the diseases that is caused is called spongiform. Actually, the whole name for all the diseases that it causes is spongiform encephalopathies, because they all make the brain looks like sponges, okay? There are different types of diseases caused by prion. Some affect these affect human. There is one that affects the cow, which is the mad cow disease, and one that affects sheep. So they all happen through something that is not even a virus, and it's even smaller than a virus because it's just one single protein that can cause all this harm. There is no treatment for this. So the only thing that really can happen is you be able to treat your cattle and sheep adequately and control the appearance of these prions. We're done with chapter 13. I'll see you soon for chapter 14. Bye.